Jai Swaminarayan to all students. As you know, in previous video, we have already discussed about pre-fertilization event of the plant. In this video, we are going to discuss about double fertilization and some post-fertilization event. First one is a double fertilization. As you know, synergid cells are two specialized cells that lie adjacent to the egg cell in the female gametophyte of angiosperms and play an essential role in pollen tube guidance and function. So, after entering one of the synergids, the pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. As you know, syngamy is the act of the fertilization. This results in the formation of a diploid cell, we can say zygote, that is 2N. You can see in the diagram, the other male gamete moves towards the polar nuclei located in the central cell and fuse with them to produce a triploid primary endosperm nucleus, that is PAN. You can see in the diagram. As this involves the fusion of three haploid nuclei, it is termed as a triple fusion. You can see in the diagram, primary endosperm nucleus 3N PEN. It is already there. Here, two types of fusion, syngamy and triple fusion, take place in an embryo sac. The phenomenon is termed as a double fertilization. The central cell after triple fusion becomes the primary endosperm cell and develops into the endosperm while zygote develops into an embryo. Now, these are the events of the post-fertilization. First one is a development of endosperm, then development of embryo, then maturation of ovule into the seed and maturation of ovary into fruit. Let's discuss one by one. First one is a development of endosperm. The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food material and they are used for the nutrition of the developing embryo. The PAN undergoes successive nuclear division to give rise to free nuclei. We can say it's a free nuclear endosperm. Here cellular formation occurs and the endosperm becomes a cellular. The number of free nuclei formed before cellularization varies greatly. If we are taking example of coconut, the coconut water from tender coconut is nothing but it is a free nuclear endosperm and the surrounding white kernel is our cellular endosperm. Now next is a development of embryo. As you know, embryo develops at the micropillar end of the embryo sac where the zygote is situated. Most zygote divides only after certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. These are the some structure. As you know that though the seed differ greatly, the early stages of embryo development, we can say embryogeny, are very similar in both monocots and dicots. Here some different shapes are there. The stages of embryogeny in a dicotyledonous embryo, it rises to the proembryo and subsequently to the globular, heart-shaped and mature embryo, as you know. Now next is a typical dicotyledonous embryo. You can see in the diagram A, it consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledon is the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyle, that terminates at its lower end in the radicle, or we can say root tip. The root tip is covered with a root cap. You can clearly see in the diagram A. Now students, see the diagram B. Embryos of monocots possess only one cotyledon. As you know, in the grass family, the cotyledon is called scutellum, that is situated towards one side of the embryonal axis. You can see the diagram B. As its lower end, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath called coryza, and the portion of the embryonal axis above the level of attachment of scutellum is the epicotyle, and epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure called a calyptile. You can see in the diagram. Now students, next is a seed. As you know, in angiosperm, the seed is the final product of the sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruit, as you know. A seed typically consists of seed coats, cotyledons and embryo axis. You can see in the diagram. Generally, mature seed may be non-albuminous, albuminous or perisperm. Let's discuss it one by one. Non-albuminous seeds have no residual endosperm, okay, as it is completely consumed during embryo development. Means non-albuminous seed have no residual endosperm. We are taking example of this type of seed is pea and groundnut. Second one is the albuminous seed. It retains a part of endosperm as it is not completely used up during embryo development. We can say it has endosperm. Examples are wheat, maize, barley, castor, etc. Now, third one is a perisperm. In some seed, such as a black pepper and beet, remnants of nucellus are also present. So, this residual persistent nucellus we can say is a perisperm. Now, the micropyle remains as a small pore in the seed coat, as you know. These facilitate entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seed become relatively dry. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, they can generate. Now next and our almost last topic is the development of fruit. As ovule mature into seed, students, you know that ovary develops into the fruit. The wall of ovary develops into the wall of fruit called pericarp. In most of the plant, by the time the fruits develops from the ovary, other floral parts degenerate and fall off. There are three types of fruit: false fruit, true fruit and parthenocarpic fruit. So, first of all, we are discuss about false fruit. In a few species such as the apple, strawberry and cashew, you can see in the diagram, the thalamus also contributes to fruit formation, and these type of fruits are called as a false fruit. Next one is a true fruits. Most of the fruits, however, develop only from the ovary and this type of fruit are called as a true fruit. Although in most of the species, fruits are the result of the fertilization, but there are a few species in which fruits develop without fertilization. Such fruits are called as a parthenocarpic fruits, example banana. So, parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Now next is the importance of seed and its viability. As you know student that we can't imagine our agriculture without the seed. So seed is the basis of our agriculture. You know, in a few species the seeds lose viability within a few months. Some can live several years while some can remain alive for the hundred of years. Here you can see some example of the seed viability. Next and our last topic of this chapter is apomixis and polyembryony. Now what is apomixis? Seeds are the product of fertilization as we know. A few flowering plants such as some species of asteraceae and grasses have evolved a special mechanism to produce seeds without fertilization. We can say it's a apomixis, and occurrence of more than one embryo in a seed is referred to as a polyembryony. So students, with this video I finished chapter number 2. From next video we will start our chapter number 3. Thank you. Jai Swaminarayan.